So
welcome everyone to the second webinar in 2023 of the Early Career Members Network of the Metabolomics Society. Thank you very much for joining us today. My name is Álvaro Fernández and I am a postdoc researcher at the University of Granada. And I will be co-hosting this webinar with Antha Ramabulana, who is a PhD student in the University of Johannesburg. The first part of the webinar will be recorded and posted on the Metabolomics Society website. We would like to encourage you to connect to our social media networks via Facebook or Twitter for future content. Feel free also to mail us for future content or any suggestion for future speakers or topics for next webinar or other input.
welcome everyone. I'm Anzara Mabrana. Our two speakers today are Professor Ntakadzeni Madala from the University of Johannesburg. Sorry, everyone. Our two speakers today are Professor Ntakadzeni Madala, who holds a PhD from the University of Johannesburg under the supervision of Prof. Ian Duberry, and he has worked extensively in the field of plant metabolomics. He is currently an associate professor in the biochemistry department at the University of Venda. His research interest is on metabolite fingerprinting of underprivileged plants by LCMS techniques to enhance the pharmacological value thereof. Recently, he has also started working on analysis of naturally occurring isomers using a makeshift UV light for photoreactors mimicking sunlight exposure of plants. He has generated new isomers, which he uses as a model for isomer identification through LCMS. His work has shown that plants contain molecules prone to UV geometrical isomerization as an evolutionary strategy to protect themselves against excessive sunlight exposure and other stressors. Then the second speaker will be Ms. Babra Moyo, who is based at the University of Venda as well. She is currently registered for a PhD in biochemistry at the University of Venda, and her research is on investigation of chemical relationships between host plant and semi-parasitic plants using the UHPLC Q12MS and molecular networking. She's also working as a lab technician in the Department of Food Science and Technology in the same institution. She has obtained a MSc in chemistry from the University of Venda and our honors in chemistry and mathematics from the University of South Africa. We look forward to hearing from the two speakers today and hope you will all enjoy the presentations that are coming. Prof. Madala, over to you.
Anza, please confirm if you can see my slides.
Yes, we can see your slides.
Thank you. Do I have to have a video on, or is it fine to continue like this?
Maybe you can just greet your audience and then continue with your video off.
Sorry, I just want to see whether if I think my video is not coming on, but it's fine. Without wasting time, let me greet everybody who make time to come and listen to us today. when we give this presentation today together with my PhD student, Babra. So the topic that we chose today was to just give an overview of the work that we are doing here at the University of Venda, when we chose the title of Overcoming the Isomer Challenge during Metabolite Identification by LCMS Technique. So I'm just going to give an overview of some of the success story that we had ever since we started working with isomers. So here at the University of Venda, our main purpose of doing this research is to enhance the nutraceutical value of underutilized plants by means of looking at their metabolite composition using LCMS. So as you can see from this slide here, many plants have been characterized well, and some of them has been industrialized. Some of them will rely upon them for many, many purposes, as you can see here. For instance, the cotton plant that we get cotton from to make clothes, some we use as medicine, some we use to control pests. So it goes to show you that plants are very rich with chemicals that can be applied in many, many fields. So here at the University of Venda, we use underutilized plants, as I've already mentioned. Some of the plants that we use as model plants are listed here. You can see Bidens pilosa, Helichrysum kraussii. Let me just have my pointer, sorry for that. Yeah, Helichrysum kraussii, you can see Moringa oleifera, Momodica, Balsamina, Viscum combreticola, which Babra is going to present on, Concenia riemanni. So these are plants that are found everywhere around this region where we are, and some of them that are used as food, some of them that are used as medicine. So here at University of Venda, we look at their chemistry in order to enhance their pharmacological value. So plants do produce a lot of chemicals. However, these chemicals are not produced for human consumption. Plants produce these chemicals for their own protection. However, we have found that we can use these chemicals for multiple purposes, such as nutrition, also for medicinal purposes. And it is well known that the medicinal properties or the nutritional value of a plant could be easily correlated to its metabolite composition. The metabolite composition can be different depending on many things. Of course, plants produce different chemicals owing to the biosynthetic enzymes, and some of the chemicals are diversified inside the plant through chemical means such as glycosylation and isomerization. In today's talk, we are going to only focus on isomerization, which I've already described for you here. That is when a molecule having the same atom, but these atoms are arranged differently from one molecule to the next. As you can see at the bottom here, this is an example of an isomer, a very complex isomer, where, for instance, you can see that this molecule has a cinnamic acid or a caffeic acid attached to this position, whereas this one, this cinnamic acid, has shifted from this position to this position here. These are referred to as positional isomers. So we are going to look at how these isomers influences the chemistry of the plant and how difficult are they when we try to identify metabolite. Because when you identify metabolite, especially when you're a biochemist, you know you have to give a definite identification because these isomers have different functions depending on the structure that they possess. So one of an example of highly isomerized molecule is chlorogenic acid. These chlorogenic acids are formed as an ester between a quinic acid and cinnamic acid. Mostly there are 4 different cinnamic acids, but these first 3 are the mostly commonly found in most plants. These acids, they esterify themselves on this OH group, thereby forming molecules called chlorogenic acids. These chlorogenic acids, of course, when the acylation happens, you can form different isomers. You can form, if it acylates here, if the cinnamic acid acylates here, you form 1 chlorogenic acid, 3 chlorogenic acid, 4 chlorogenic acid, 5 chlorogenic acid. These compounds have been found to have multiple biological properties or functions. For instance, they are known to be anti-diabetic, some are known as antioxidants, and some are known to bind HIV integrase enzyme. The presence of these isomeric compounds, however, poses a very interesting challenge, analytical challenge. And we found ourselves in laboratories trying to find ways in which one can distinguish between these isomers. Of course, mass spectrometry method has been found to be superior. And in 2003, Clifford and colleagues have developed an ion trap-based mass spectrometry based on multi-stage fragmentation in order to distinguish between these positional isomers. So because of the multiple positions or multiple OHs, sometimes you find that more than one cinnamic acid acylates the quinic acid, thereby forming, you can either form monoacyl if one acylate, or diacyl if two acylates, or triacyl if three acylates. The numbers in brackets are just the number of positional isomers that are likely to form if you have mono or di or tri. So other than the positional isomers that we know, plants, as we know, they are constantly exposed to direct sunlight. We as human beings, when the sunlight or the sun is too hot, we can run under the shade. However, plants cannot do that. Well, how do they mitigate the effect of sunlight or excessive sunlight? These plants, through this cinnamic acid, they undergo what is referred to as geometrical isomerization. Due to this double bond, which is naturally existing as a trans, when it's exposed to light, it changes its configuration to cis, thereby amplifying the already complex metabolome of plants, which it is populated by positional isomers. Now, on top of positional isomers, now we have geometrical isomers. And due to ever-changing climate, there's excessive sunlight reaching Earth's surface. And these are leading to metabolite shift, whereby now we are seeing a lot of accumulation of these geometrical isomers in multiple plants, which were not there before, because, as you know, with global warming and climate changes, there are a lot of sunlight reaching Earth, thereby changing the chemistry of plant. We'll see how we mitigate this analytically. So we have now posed several questions, and these several questions comes from, for instance, analytically, to say, how do we develop techniques that are able to comprehend this positional and geometrical isomerization? Secondly, what are the biological consequences of this geometrical isomerization? I put one question in red here, where I said sunlight or UV light induced geometrical isomers, are they good or bad? These compounds are new, and no one knows the implication of their presence. Up to this point, I think we are the only research group who are extensively publishing on these molecules, and we are trying to look at them from different perspectives in terms of their biology, in terms of their identification analytically. So in order to understand this molecule, or to analyze this molecule, we started by optimizing chromatographic methods in order to see whether if we can be able to rely on chromatographic elution order. As you can see from this slide here, that using different columns, for instance, using a bifinal column versus a C18 column versus another C18 column versus another bifinal column here, you can see that the order of elution is not consistent. For instance, in the top one here, you can see 4 isomers. Here you can see 4 isomers, but the order, as you can see on this C18, it changes on another C18, which therefore means one cannot rely on chromatographic order in order to identify these molecules. So what is next? Then we opted to go for mass spectrometry. One of my former master students worked on this molecule extensively, and she has found that some of these cis isomers that are formed post UV exposure, the tall one here is a trans isomer, and the rest are formed as consequence of UV exposure. They have the same mass, they have the same fragmentation pattern, which makes it difficult for us to distinguish between them. As you can see here, we cannot even use chromatographic order. Here in the next step, we try to use mass spectrometry, and we found that using mass spectrometry, we can be able to distinguish them based on adduct formation. We found that the cis isomers, because of their shape, they are able to accommodate alkali metals such as lithium, sodium, and cesium. And we saw that they chelate these metals better than the trans, which therefore means one can distinguish between trans and cis. But as you have seen in the previous slide, there are 3 cis isomers. Here, now what we can be able to do is to tell which one is trans, which one is cis. But between the cis, we couldn't distinguish between them. So the fact that these molecules bind to metals I have now a collaboration with Dr. Murlan, who's my colleague here, who we are looking at the potential of these molecules as corrosion inhibitors, because you know in corrosion, you want something that can bind a metal so that when a corrosive environment comes, the metal is protected. And we can say now that we have proven this hypothesis that the ability of these cis isomers to bind metals can allow us to apply them industrially in order to prevent corrosion of steel and other related metals. So now we moved on to use ion mobility mass spectrometry using the drift tube ion mobility in collaboration with some colleagues from the United States in order to see whether if we can be able to distinguish between the cis isomers that are formed. And with the aid of computational modeling and ion mobility mass spectrometry, we managed to identify each newly formed isomer with high degree of confidence. So this ion mobility mass spectrometry combined with theoretical calculation was enough for us to be able to distinguish between different cis isomers that were formed post UV exposure. So this is just a schematic representation showing the different positional isomers of dicarphyl-quinic acid in the 90 centimeter ion mobility drift tube. We could separate them to a certain extent, but the separation was not good. But when you use a longer drift tube, you can see that indeed the separation was efficient. And moreover, when we then used another technology called SLIM, we could be able to distinguish between the isomers. And again, we show that these are better off you can better separate them if they form adduct with sodium. And as you can see here, this is non-UV exposed here in a 90 centimeter drift tube ion mobility spectroscopy. You can find 3 peaks, which the center one you couldn't resolve. But when you use SLIM, you can see that now you are able to resolve them. And we found that our theoretical prediction of the collision cross-section was correlating very well with those that were determined experimentally, which then shows you that we can be able to tell using ion mobility which isomer is which. So what is the biological consequences of this molecule? The biological consequences of this molecule, we saw it in this study in one of my former master students who looked at a plant that accumulates a lot of these isomers, which was growing naturally. And she has found that all the isomers, regardless of their geometry, whether they are trans or cis, they participate towards mitigating the effect of gamma radiation, which is oxidative stress. And here we found that these molecules, regardless of the geometry, they do participate in the oxidative stress induced by gamma radiation, which therefore suggests that the presence of geometrical isomers is not just a mere structural artifact. They have a biological role to play in a plant, and the existence of this large pool of polyphenolic compounds, which compose of trans and cis, contributes to the state of readiness, meaning that the plant that has more of these isomers is more ready to defend itself against stress. And this we termed it, or it was previously termed it, as better be safe than sorry. So the plant accumulates both positional and geometrical isomers so that when stress comes, it can use this as a chemical arsenal against the stress. So we also looked at the binding of these newly found isomers to HIV-1 integrase enzyme, and we find that indeed these isomers do bind to HIV enzyme, and then they do occupy the active site of the HIV-1 integrase enzyme. As you can see here with the bold amino acids are those that makes up the catalytic site or the active site of the HIV-1 integrase enzyme. So this is just a ribbon structure to show how they bind, and some of the isomers were found to also bind to the magnesium cofactor, which therefore again inhibits the enzyme, which therefore means that if you use a plant that has accumulated geometrical isomers, the likelihood is that you're going to have more isomers that can occupy the active site of HIV-1 integrase enzyme or with multiple poses, thereby inhibiting it. And of course, together, they can confer a synergistic effect, just like what we saw in the plant that was experiencing oxidative stress in when exposed to gamma radiation. We then went on to work on similar molecules, similar to chlorogenic acid, which is referred to chichoric acid. This is the same as chlorogenic acid. The difference is that the anchoring molecule is no longer chichoric acid, but so it's no longer a quinic acid, but it's tartaric acid. Here's the trans, but when you expose this trans to UV light, of course, these arms can change from trans to cis. In this case, either one of them can go cis, then you call it a monocis. Because they're symmetrical, you cannot distinguish between them. So you form one cis. On this other one, you form two cis when the both arms changes to cis, then you form a dicis. So instead of having four isomers, like in the case of chlorogenic acid, here you form a total of 3. So when we did the same thing, when we separate them using different columns, we use the C18, phenyl hexyl, biphenyl, and PFFP. We saw that the best performing column, which can show us all 3 peaks, was the biphenyl column. And then the other ones couldn't show. Like for instance, if you use this, in this panel, we use methanol. In this panel, we use acetonitrile. If you use methanol and C18, you only see just 2 peaks. If you use acetonitrile, of course, there's the emergence of the other one coming out of this one, which here it just looks like a perfect peak, but here it shows that it's splitting when you use acetonitrile. Same thing here is more pronounced when you use acetonitrile. Here with methanol, very pronounced. Here also you can see it with acetonitrile. But you can see now the order of elution when you use the PFFP column. You can see that now the cis isomers are coming before the trans. That means the order of elution cannot be relied upon. But when you put it in MS, we use a triple quadruple instrument here. You can see that the ratio between the peak of tartaric acid to that of caffeic acid changes as you move from trans to cis. You can see that here the base peak is 179. Here the base peak becomes 149. And it's even more pronounced when both arms become cis. And this can be a premise that one can distinguish between geometrical isomers of chichoric acid. Unlike on the chlorogenic acid, where we have to rely on metal binding, here naturally these molecules, if you fine-tune the collision energy and other fragmentation energies, you can be able to distinguish between the trans and its cis isomers. So again, we went on to investigate 2 plants that are eaten as food here in the Venda region, and we found that the 2 plants, which is Bidens pilosa and Soccus oleracea, produce 2 different chichoric acid. For instance, I've told you the arms of the chichoric acid, the 2 caffeic acid, are symmetrical to one another because you can see the bonds here are the same. However, if you look at this one here, you'll see that the bonds are not symmetrical, which therefore means this one, when you form the monocis, whether you change this side or this side, you cannot able to distinguish. But on this one, you'll be able to distinguish. And then we saw that these 2 plants produce different chichoric acid because when we analyzed them on chromatography, the 2 peaks representing chichoric acid were appearing at 2 different rotation time. Now, as we move on, we saw that using computational modeling with a friend of mine, did a modeling using DFT, that the RR chichoric acid, which is the one that is symmetrical, has a different shape from the one which is RS, and hence, due to the presence of intramolecular hydrogen bonding, the polarity of these 2 molecules is different, hence they appear on different rotation time on the chromatography. We then went on to look at their binding on the HIV-1 integrase enzyme, and we found that the RS conformer, which is the MS, or which is found in Bidens pilosa, binds more strongly than the RR, which again confirms the use of, or validate the use of Bidens pilosa in these regions by HIV-infected people, where they say indeed it offers or it gives them a much relief to the viral load. There was even one guy who came to my office one day and says he uses it and he goes to the clinic and check his viral load, and then he found that it's very low. Maybe this could be the reason. We just demonstrated using computational modeling to show that the RS, which is found in Bidens pilosa here, it is more potent than the RR1. The other isomers that we are studying in our research group include, amongst others, chlorpamide, which is found in a plant called Velonia fastigata. This one, we have shown that it can form geometrical isomerization, for instance. Here, you can see this is a natural plant. We analyzed it on MS, and we found that there is the trans, and there's an emergence of the cis in naturally growing plants because they're exposed to sunlight. When you take the extract and expose it to UV light, you can even generate more of this cis isomer, and which is here slightly equal to the trans, which means that artificially one can generate these isomers by exposing the plant extracts to sunlight or to UV light. Another example which we studied was the plant called devil's claw, which is used as an anti-inflammatory plant, and it also contains these cinnamic acid containing molecules. When you expose them to sunlight, you can see that the number of peaks increases from the two that were here. Now we can see more are actually coming down here. So now we went on to also study another plant compound containing cinnamic acid called verbascoside, which is found in Lipia javanica, and we found that these compounds also, they undergo geometrical isomerization post UV exposure. From this one, the chlorpamide, the hepagyne from devil's claw, and the chlorpamide, the verbascoside from Lipia javanica, we couldn't distinguish them by using mass spectrometry because they fragment exactly the same way, and they don't form metal adduct or formic acid adduct that we can exploit in order for us to be able to distinguish between them. So we then decided to move on to look at the whole plant and not look at the single molecule and see what would be the effect of exposing plant extracts to UV light. And we can see here, using an example of some of the compounds that we know to contain cinnamic acid, here's the combination of them up here in a naturally growing plant. When the plant extracts of the same plant were exposed to UV light, look at the number of peaks that become to existence or they emerge. So what we then decided to do is to look at tools that we can use in order to visualize these newly formed isomers. So we opted for molecular networking in one of the plant that we studied with one of the PhD students who was looking at different net shading. So she was looking at the fertilization of bush tea and using different net shade that can filter sunlight in order to see whether if the chemistry of the plant will change, especially this cinnamic acid containing molecule, will they change if she can filter the sunlight that reaches the plant. And indeed, using molecular networking, we can show that yes, there are compounds in bush tea that are susceptible to UV light, and net shading does influence the chemistry of the plant, which therefore, again, we can say it can influence the biological properties of that particular plant. So one of my master students who recently handed his master student published a paper in the South African Journal of Botany, which sums it up all, that ultraviolet light-induced geometrical isomerization of cinnamic acid-containing molecule is a plausible, non-enzymatic approach to modify metabolite composition of plant extracts. This method, which we showed here, shows that indeed, when you take a plant extract, expose it to UV light, newly formed isomers that have same mass, of course, same fragmentation pattern, of course, sometimes with slight different hydrophobicity that we can see on a chromatogram when they appear at different rotation time. It is it's a useful method to generate new isomers. What those isomers do from biochemistry is a very interesting question, and we need to find ways in which we can apply them in biological sciences, in industrial processes such as corrosion inhibition, to see whether if they can offer any function, because these isomers are becoming part of plant chemistry, as we have demonstrated with these slides here, that they are there, and they're there, they will always be there, and as such, we know that we need to find ways in order to identify them. Hence, we opted for LCMS, and in our method, we can see that indeed LCMS is a superior method. We analyze these extracts on LCMS. As you can see here, this is a non-exposed plant extract. Look, when you expose it to UV light, newly formed isomers are formed. And these isomers are many. Of course, I said they change in hydrophobicity, and then we can be able to quantify this change in a whole plant using normal PCA and cloud plots. You can see at the bottom, newly formed compound due to UV exposure. And as I've said, this is artificially done in the laboratory because we have an instrument that we can take plant extract, expose them to UV at time intervals, and be able to generate these isomers and study them. But now the problem is we come from a very sunlight extensive environment where our plants have these compounds naturally. Even if I go out now, pick up the plant, come and extract it and analyze them in the LCMS, I'm guaranteed that I'm going to see geometrical isomers. So it is therefore important that we develop analytical means to comprehend these isomers and understand them and be able to find ways in which we can identify them, because we cannot just identify one and say this is an isomer of this, this is an isomer of this. Biologically, it does not make sense, because you might find that of all these isomers that are appearing at the bottom here, only one of them is active. Therefore, it is important that we must develop analytical method that can allow us to identify this molecule with high degree of confidence or high degree confidence. So therefore, it is important that we develop LCMS methods that are able to do this job. So as I conclude my presentation, to give Babra a chance, I will say natural products are very attractive analytical models, and the presence of isomers poses a serious but interesting scientific challenge. We do spend time with my students figuring out these isomers, creating new ones, studying them with LCMS to preempt what could be happening in the environment. And then we can see from our LCMS results that the MS is more superior in distinguishing these isomers, and one the method from one type of MS, like in the case of ion trap mass spectrometry, can be also be applied with QTOF, and you can still attain the same identification capabilities. Plant metabolomes are complex, and environmental changes such as those associated with climate change are resulting in emerging isomers. These are the geometrical isomers now, of which the biological consequences are still unknown. We're investigating that in our laboratory. We're looking at how these isomers influences the entire chemistry or biology of the plant extract. The presence of geometrical isomers in natural plants, that means those that are growing in an environment, allows an opportunity to rationalize future drug design characterized by minimal chemical modification. For instance, I have a colleague here who's conjugating now cinnamic acids in known drugs. And what she does with those, that means when she UV expose them, she can form geometrical isomers of the cis isomer that will be formed after UV exposure. And what we noted is that using HPLC and LCMS, the changes in hydrophobicity of the entire drug changes just by mere change of the geometry of the cinnamic acid. So that means these cinnamic acids can be used as tags to change the hydrophobicity of known drugs. And remember, cinnamic acid on its own is a biological active molecule. That means you're gonna have a drug that has both active site and the cinnamic acid, which can offer you a synergistic effect. Of course, you can do polarity switching. And again, because of the geometrical isomerization enabling this molecule to bind metals, you can now create pro-oxidants, molecules that can bind metals that are known to be pro-oxidants in a physiological environment. So that means by limiting metals such as copper, you can then now form molecules that can act as an antioxidants. So I just put a picture of one example here, which is belorabine, which is a molecule that has been made, and it does contain a cinnamic acid derivative here. That means one can change the geometry of this molecule. Therefore, by changing the polarity of the molecule, we don't know whether if that affects the biology or not. But as far as we have seen currently is that because of the presence of all these isomers, it's better to have more because they operate synergistically. I want to thank my collaborators from University of Johannesburg, Dr. Fidel Tugizmana, Mzizi, UKZN. We have Professor Mdau, University of Surrey, I've collaborated mostly with Mike Clifford, who was the first person to develop a method to look into the fragmentation of chlorogenic acid. All these people that are listed here are people that I'm collaborating with. The students are also thanked for the work that they do for us, and then we can be able to give presentations like these ones. I'm grateful to the funding that I got from the National Research Foundation of South Africa, University of Venda, my time at University of Johannesburg. Currently, I have some funding and fellowship with the Stellenbosch Institute for Advanced Studies, and Shimazu, which is the main supplier of our instrumentation. They also supply some funding, seed funding, that we do some project for them, and also which allows them to use our results to market their instrument. With that, I thank you all. Thank you very much.
Thank you so much, Prof. I encourage everyone to be posting questions in the chat box, and then we'll